Well, good morning, and welcome to the third biennial Osher Center for Integrative Medicine Network Forum. Thank you all for braving the weather to be here today with us. I'm Peter Wayne, and I'm the Research Director at the Osher Center, and I'm pinch hitting as the program MC for Dr. Helene Langevin, our Director, who unfortunately could not be here today for reasons she'll explain to herself in a short video that she's recorded for us. But before sharing Helene's video, I want to say how thrilled I am with the exciting and timely topic for this year's forum, which aims to fold the field of integrative medicine into critical discussions regarding how much of our health is controlled by nature, that is, by our genes, and how much by nurture or our lifestyle, and how better understanding this can inform and better situate integrative medicine's contribution to the emerging paradigms of patient-centered care and personalized medicine. I'm so pleased as well by the shared enthusiasm for this topic within our community. At last count, I think we had close to 450 registrations, including guests from as far away as Korea, Brazil, and India. So thank you all for coming. In addition to having the opportunity to learn directly from our outstanding invited speakers, and especially our keynote, Professor George Church, I hope you take advantage of this forum to network with others by visiting posters, participating in our breakout groups, and making new acquaintances during our breaks. So before moving on to the main part of our program, Dr. Langevin wanted us to share a short video message with you that she's pre-recorded.
Good morning, and welcome to the 2018 Integrative Medicine Network Forum. It is with some sadness that I am speaking with you all on video rather than in person, as I had planned. I have been selected as the next director of the National Center for Complementary and Integrative Health at the NIH, and I will begin in my new role at the end of the month. I was asked to give a presentation today at the Interagency Pain Research Coordinating Committee in Bethesda, and this is why I am unable to be here today. I am, however, thrilled to introduce our third Integrative Medicine Network Forum. As in our previous two events in 2014 and 2016, a main goal of the 2018 forum is to convene the Boston integrative medicine community and provide a platform for presenting the exciting work that is happening throughout our research and clinical networks. Today's theme of genetics, epigenetics, and lifestyle addresses the fascinating relationship between our hardwired biological processes and how we live our lives. I invite you to enjoy this morning's presentations and discussions and take advantage of the networking opportunities. It has been very gratifying to see the growth of the Boston integrative medicine community over the past 6 years. 6 years ago, when we first built our network maps, we wondered what happens to a network when it becomes aware of itself. I invite each of you to reflect on this throughout the morning and think about what it means to you to be part of this integrative medicine community and what you can do to make it even more vibrant and connected. The forum is just one vehicle through which to engage. I hope all of you will continue to participate both online through our network maps that you will learn more about today, and by attending our monthly Grand Round series, which have been one of our contributions to providing continuity for the integrative medicine community between these large biennial conferences. Although I will miss all of you, I am happy to be leaving the Osher Center in excellent hands, as Dr. Peter Wayne is taking on the leadership as Osher Center Interim Director. I will now say goodbye and hand the podium over to him, as he will introduce our keynote speaker.
Well, I know many of us here today are really sad to be losing Helene's remarkable leadership. We will miss her fierce intelligence, her inspiring vision, and for those of us who got a chance to work with her, her kind-hearted and collegiality and friendship. However, I know we're also very happy for her to have this distinguished opportunity at NCCIH, and NCCIH is very lucky to be having her lead their mission. I can't think of anyone more qualified for the task. I have no doubt, in addition to having great successes within NCCIH, she's likely to infiltrate and impact all 27 institutions and centers at NIH. And as she did at Harvard, take down silos and serve as a catalyzing force for a more integrative, creative, and promising future for medicine as a whole. So I want to take a moment just to salute Helene. Just one more announcement before introducing our keynote speaker. So in an attempt to create a more engaging and efficient method of fielding audience questions for our speakers today, we're experimenting with a new app called Slido that our guru, administrative assistant Jessica Kaufman here has brought to the table. The app allows us to post questions for our speakers during and right after their presentations. So for those of you who want to participate, please take out your cell phones. That's an unusual question in an audience like this. On silence, of course, or your laptops, and I'll walk you through a few simple steps that are involved. And I think, yeah. Okay, so first, you just need to go to slido.com using your browser. Okay, and I'll give you a second to do that. So when you get there, you should see a text box where you can enter the event code, and the event code is OSHER. It's not case sensitive, right? Not case sensitive. So if all goes well there, you'll see a practice poll. And we created this poll to make sure that Slido works for us here today. So in the poll, you simply select your answer to the question, have you previously attended an Integrative Medicine Network Forum in 2014 or 2016, and then click Send once you've done the answer. Wow, it really works. And we get to see that we have a lot of newcomers here, which is fantastic, but we also have a couple frequent flyers, which is fantastic as well. And I think the numbers will keep populating that. Okay. So during our talks, if you'd like to ask a question via Slido, simply return back to this page, click on the option for questions, and then enter your question. And you'll have an option to either put your name there or to remain anonymous, and then just simply hit send. Your question will appear for everyone else in the audience to view, and because of that, you'll have the ability on your phone to upvote questions by clicking the thumbs up. And that'll elevate the questions that are of most interest to the audience to our panel discussions. So we'll collect your questions throughout the keynote presentation, so there won't be questions after that, as well as during the panel. And then these will be selected by our moderator, Dr. John Denninger, to field and pass on to our panel discussants. So it's now truly my distinct honor to introduce our keynote speaker for today, Dr. George Church. Listing Dr. Church's accolades and accomplishments could take a full hour or more, so I'll be brief here. Matching the integrative and transdisciplinary mission of the OSHER Center, Dr. Church's contributions uniquely intersect biomedical, technological, ethical, and social issues related to genomic science. Among his many current titles is that he's a professor of genetics at the Harvard Medical School, a founding member of the Wyss Institute, and a director for an NIH Center of Excellence in Genomic Sciences. He's widely regarded as a pioneer in the fields of genome sequencing, engineering, and manipulation, and his innovations have been the basis for a number of highly successful and impactful private companies. His efforts have led to over 450 peer-reviewed articles and 105 patents, and he's the author of the provocative and visionary book, Regeneration, or How Synthetic Biology Will Reinvent Nature and Ourselves. So until this morning, I've never had the opportunity to personally meet Dr. Church, but I've felt this sort of kindred connection as he completed his PhD in the Department of Evolutionary Biology in his lab on the same floor as the one I spent 15 years in doing my research in evolutionary biology before making the shift. So I'm really personally excited that today's program will bridge ideas related to evolutionary biology and integrative health. So without any further ado, please join me in welcoming Dr. Church.
Thank you very much. It's really a great pleasure. I can't think of a topic that I am more excited about and dedicated to than integrative health, integrative medicine, and certainly epigenetics and lifestyle are a key component of that. These are some of my conflicts of interest and also some of my thank yous to nonprofit and for-profit that help us get our crazy ideas into social applications. Now, I actually mean that when I say this is future of medicine, even though it's a kind of an ironic slide that I lifted from the National Cancer Institute website. Here it shows their pie chart, and prevention is the little 1% black sliver on the right-hand side of the pie chart. That's how much they spend on prevention. And they spend a lot on, powerful small molecule drugs like the one here, relapsing after 9 weeks. So we'll get back to prevention in just a couple of slides, but I just want to mention where I come from, which is technology. I'm mainly a technology developer, and the technologies I focus on tend to be a broad scope, highly disruptive, and so-called exponential, meaning that they change by factors of 10 per year, so that we get, for example, a 10 million-fold change in our ability to read DNA. That's in cost, and then the price is typically more than the cost until this year, where now the price is less than the cost. The price as of yesterday at Nebula Genomics is $0. So we are offering the first $0 human genome just not that many years after we supposedly finished the first $3 billion genome that was much celebrated, but I've been very critical of before, during, and after the project, because it was not only too expensive to be equitably distributed, but it also was a very poor genome from a clinical standpoint. To get your clinical genome, you need to be able to tell your mother and your father's contribution separately. Excuse me. And the genome project was aimed at a consensus sequence, a single genome. So it was clinically completely inadequate. But the one that we're doing now, Veritas has been offering for less than $1,000 for 3 years, and then in collaboration with a new company, Nebula, $0. Now, why were we able to do these improvements, these exponential improvements? And I would argue it was not automation or miniaturization or scale-up of existing technologies. It was completely rethinking it in terms of multiplexing. Now, multiplexing sounds like jargon, but Edison invented it in the 1800s for telegraphs, and we still use it for communications. And the molecular multiplexing means that if I just take a tube, a standard test tube, little microtube, it can do billions of reactions with the same effort that it used to take to do one reaction. So it's a billion times easier without any particular, you can add automation to that, but the key thing was multiplexing. So back to prevention and cancer. So the NCI spends 1%, but the return on investment is incredible. We have all these ways of preventing cancer that are quite powerful in extending quality-adjusted life years. We can do preconception genetics. We can do adult genetics where you take preventative action, like removing parts of your body, some of which you would prefer not to, but it avoids decades it adds decades of life. Avoiding environmental factors, and even infectious diseases are a great source of cancer and are preventable for things, for example, the HPV vaccine. Now, to be more mindful of our environments and to have a more integrated health view, we need better environmental sensing. In fact, I would say our society is almost phobic of decent diagnostics. We have had the technology for a while, but it is very rare for someone to get, for example, if you have a cough or a respiratory infection, or it could be allergy, it could be just about anything. We don't diagnose it. It's very, very rare. I insist on it for myself, but it's extraordinary. Anyway, there is now a handheld. 2 different companies offer nanopores, at least, but one of them is is handheld, Oxford Nanopore, and it has the record for the longest read lengths as well. So sequencing is how long you can read before you kind of run into a mess. And this you can read hundreds of thousands of bases, and you can do it in minutes after sample. So I think that this will allow us to monitor our environment, not just for DNA, but for a variety of other things as well. Now I'm going to give 8 quick examples. These are going to look a little technical, but this message is very simple, and I'm going to present each of the 8 examples in a simple form. But 8 different ways that we have aging reversal. When I use the term aging reversal, people immediately say, oh, science fiction, and it's not. These are 8 things that have been tested in animals, typically mice, and they're quite dramatic. And I've taken quotes from articles here and given the references so you can judge for yourself whether these meet your criteria for reversal. But the first category are two drugs, small molecule drugs, like small molecules like aspirin, so where you can draw the structure on the corner of the slide. And these examples are rapamycin and metformin, and you may have heard of these if you're aging buff. And they work in the AMPK mTOR pathway shown there. Doesn't matter. The fact is there's a lot known about the molecular mechanism. They're being tested either in mice, dogs, and humans. Second example, completely different, not small molecules anymore, but now transcription factors. These are things that regulate gene expression. This got the Nobel Prize for Yamanaka being able to reset any cell, almost any cell, to a very early embryonic stage. So you can basically reset every major molecular marker to not just to a little younger, but all the way back. And then you can do this in vivo in living mice. So it's not just some trick you do in tissue culture. And our collaborator in the Bonvanti lab did this in 2016, and we're collaborating with them further on extending that to something that could be turned into a clinical product. That was a mouse experiment. Telomerase, probably you've all heard of telomerase. This is something where if you do it right, you can get reversal of tissue degeneration and other delays of aging in a way that does not increase cancer. You're playing fast and loose with telomerase if you don't take other precautions like messing with P53 and P16, but suffice it to say there are recipes where you can get a net increase in longevity as well as reversal of aging. Fourth example is getting rid of senescent cells so you can make them die rather than hang around and cause trouble. That's a simple concept that many people are excited about. Fifth example is another kind of repeat. The telomere is the repeats at the ends of your chromosomes, but you have repeats scattered through your chromosomes that are implicated in aging, and these have been found, if you can inactivate them, they have impact on senescence. They quote, reinstate self-renewal. Example 6, this is another famous one, which is animal-specific, where it came from, but it inspires us to move this into humans. This is the regeneration of amphibia in general, axolotl in particular, does relatively rapid regeneration where it de-differentiates into an early stage and then goes forward. Another case that can be done was historically done by suturing together a young and an old mouse, here Y and O, and then noticing, and this has been repeated in many different laboratories, not clear which molecules are involved, but the phenomenon of young blood being rejuvenating is quite clear, and it gives us an opportunity of purifying the factors, both small molecules, large molecules, and cells. And the final example is NAD pathway. Nicotinamide is a very fundamental part of redox and other phenomena that have nothing to do with redox. And mitochondrial function, and we chose as an example, most of what we're doing on aging reversal is using very powerful medicine, maybe too powerful, which is gene therapy, And so we did gene therapy on TFAM, which is in this pathway. It's a transcription factor. I mentioned transcription factors earlier with respect to the producing embryonic-like cells. Here we can make artificial transcription factors that control a transcription factor. So we can take this thing called CRISPR that some of you may have heard of, and we can turn it from a killing machine into a gentler activator. So we knock out its nuclease activity, and we instead make it so it activates a nearby gene. In this case, it's activating TFAM. And TFAM is responsible for NAD. So NAD drops by about twofold during aging of humans and mice, and we want to boost it back up at least twofold. And here we show that we can boost it by about eightfold. We can boost the messenger RNA itself, depending on the site for the CRISPR Cas9 binding, up to 48%. But it shows there's huge variation from site to site, which gets us to the topic of what's wrong with CRISPR and how do we deal with things like variation and activity from site to site and on and off target. Now we're very proud of CRISPR as a technology, but it has limitations. Some of these we overcame in our very first article, including this addressing on and off target issues. But there remains much to be done. Some of these remaining challenges, so there are errors, challenges, however you want to talk about it. I would say the main ones are number 4 and 5, which are on and off targets. But there are other ones. So, for example, using it as a therapy puts it in the category of gene therapies and orphan drugs, which are a blessing in a way, but they also are very challenging for equitable distribution. And so they don't really meet my criterion yet of being something we can get like nebula genomics, where we can get diagnostics close to $0. We would like to be able to get therapeutics to that. Now, in order to do that, in order to reduce this expensive level, there are two alternatives. One is many things that one does with gene therapy could be done instead with genetic counseling. Gene therapies cost about $1 million a dose, while genetic counseling is in the hundreds of dollars and dropping, and increasingly powerful user interface tools as well. So, but the other way to drop costs of drugs in general is what? Is to have a big market. So if you have a large number of people, so you have kind of fixed costs of research and development getting FDA approval through phase 1, 2, and 3. But if you can do that over a large market, then you can have a very inexpensive drug. And we have plenty of them that are inexpensive even before they're generic drugs. So that's where we're heading. And of course, the biggest market of all is aging. As we get more and more of us living in industrialized cities, it's now somewhere between 57 and 80% of the world lives in these cities. More and more of us are dying of diseases that do not kill 20-year-olds. So I'm going to just give two quick examples of what we're doing with CRISPR, even though I've been criticizing it right and left. I feel that I'm obliged to do that. And here are 4 different ways that we think we can beat CRISPR. We can get beyond it, and the details are not particularly compelling unless you're a connoisseur, and I'm leaving this up here so if you are. But we want precise editing. CRISPR is basically a very blunt axe that is used to kill viruses by nature, and it's used to kill genes by gene engineers. But these are more nuanced and finessed ways of integrating or removing bases and replacing them very precisely. The tau deaminases, chemical targeting, and lambda red. So those are 4 examples that are works in progress, but I'll give you some examples of where CRISPR is already, not just CRISPR, but other axes, other killing machines have been used to, it's called editing. I think it's really genome vandalism, but gene editing started with zinc fingers and talons before CRISPR, and they have been used, all 3 of them have been used, what I think is one of the most important applications, which is CAR T cells, making them universal so that you can take a large population of T cells and treat any person, not just the person from whom the T cells came, for cancer. And it started with B-cell malignancies that shared a common target, but now it's being used for a whole variety of cancers. And in fact, it's not clear that there's any cancer that doesn't have some kind of surface molecule that could be used to target, where the chimeric antigen is basically like an antibody you grafted onto the T cell. That's just recombinant DNA. But then you use either zinc fingers, talons, or CRISPRs to edit the parts of the T cell that make it hard to integrate with the immune system of the recipient. This little girl was one of the first recipients of one of the first editing molecules, which, as I said, were initially zinc fingers and talons. Now, so that's one example. The second example is related to that, is if you can transplant T cells from any person to any other person without necessarily matching them the way you did, by, say, knocking out HLA and adding HLA-E, you can do the same thing for animals, because just like 2 humans are incompatible, animals and humans are incompatible. There's a few more things to do, but it's not daunting. The part that was daunting about pig transplants, I mean, a great anatomy, quite a bit of experience in surgery as well, but they released endogenous retroviruses. We found that we had up to that point only done 2 genes at once by editing, and this required 62 changes in the genome simultaneously. And it turned out it was quite easy. We did it in about 2 weeks. It had been a problem that had been plaguing the field for about 20 years. And as far as I can tell, the FDA is pretty excited about this sudden progress. And we did it again. We've done it multiple times now on multiple strains of pigs. And there's an example of a live pig that's had all of its endogenous retroviruses removed. Now, to do that, this is not a complicated, or I'm not going to deal with this as a complicated slide. This is a very simple one, which is that to knock out genes with these genome vandalism tools is toxic to the cells. But you can inhibit it, and you can make it so that almost all of the cells are edited at every single, you can have dozens of simultaneous edits, and that's toxic unless you give it these 2 factors. And now you can see in the lower right, you get 100% have been edited at every single virus in the cell simultaneously. We've extended this to other categories of repeat sequences. I've mentioned some of these repeats already earlier in the aging category, telomeres, in particular signs and lines. They have implications for senescence that I showed some articles before from other labs. This is work from our lab where we have used this, we've emboldened by our ability to go from 2 changes in the genome to 62. We decided to go for 10,000, and we now have done 10,000 changes. This is not published, but we've done 10,000 changes simultaneously in human cells. In one cell, 10,000 changes, all targeted to one of these line elements involved in senescence, where we've created stop codons in the middle of one of their key proteins. Which brings me to one of the reasons I'm interested in organs, which is a little less obvious than the millions of people worldwide that are in crisis, in need of organs. That's quite obvious. Instead, what's interesting, the other slightly longer term thought, is that we can make organs that are enhanced. It would be very difficult to take a normal human and enhance their organs so that they could be a transplant donor. That would be unethical at so many levels. But if you're going to get an organ anyway from an animal, you want that to be as good as possible. You want, if you're dying of an infection in your liver, you don't want the incoming pristine liver to immediately get the same disease and decay itself. It turns out that pigs are resistant to quite a few human-specific pathogens like hepatitis, and so that's one approach. We have other approaches to reducing pathogen because we can engineer the germline of pigs. We cannot currently engineer germline of humans, and this is a great way of getting practice. We can make them cancer and senescence resistant. I showed you some experiments where we can make mice cancer and senescence resistant. We can make them immune compatible, and there are even 3 examples here of animals that are resistant to freezing of their whole body at once. So the idea of freezing individual organs should not be too foreign if we adapt the attitude of synthetic biology inspired by natural biology. So I mentioned the eight different ways, specific articles where aging reversal was part of their message. This is a more general list of 10 different ways of attaining longevity or aging reversal, where specifically my group is looking at aging reversal rather than longevity, partly for the very practical nature that to extend your life by 30 years, I have to do a 30-year clinical trial, which is prohibitive. But to do reversal, we can show reversal in mice and dogs and humans in principle in weeks. And that makes it easier to get, probably easier to get regulatory approval as well. So these are some of the pathways, and down at the bottom are 5 different diseases that we've tried so far. This list is growing every few months we add. But so far we have a gene therapy that is under peer review right now that addresses all 5 of these simultaneously. So 5 of the 5 that we tested. So it wasn't like there was a 6th one that we didn't succeed on, that were sweeping under the rug. This is 5 out of 5 so far, and we're open to suggestions as to which number 6 and 7 we should try with this therapy. And this therapy is based on adeno-associated virus delivery of combination gene therapies. There's not that many combination gene therapies. There's plenty of combination therapies, but these are gene therapies. And this is some of the team that is both improved AAV as a vector. They've reduced the immune. It's already the lowest immune reactivity of any of the gene therapy vectors, the ways of delivering gene therapies. This is the lowest immune reaction. The high immune reaction actually killed somebody in around 1999, Jesse Gelsinger, and caused a much more conservative approach to gene therapy since then. And now it's rebounding and is very very much embraced by the medical community. But anyway, we've made AAV better both by changing its capsid and by changing its nucleic acid to deal with innate immunity, both the humoral and the innate immunity parts. And that's work of Eric Kelsick on the humoral immunity, Kai Chan on innate immunity, and Noah Davidson on the aging reversal therapies themselves. And they all were date back to my interest in aging goes back decades to Pedro de Magalhães up there in the upper right, who as a postdoctoral fellow got me excited. We did some theoretical work and some aging gene database, and his database now based in Liverpool is probably the world's standard for looking at aging in humans and in animals. And I haven't really shown that many examples of experiments, but most of the experiments we've done on human cells are done using this cohort called the Personal Genome Project, where we have nearly annual GET conference, which stands for Genome, Environments, and Traits. And the point is the data from this project is, as far as we know, the world's only fully open access human data sets, which are big data on individuals, highly integrated on individuals, where every kind of data ranging from the tiniest molecular data to whole body, whole person interacting with their community and their environment level analyses. And it's basically, it's your cohort. Anybody can use it. It's like Wikipedia. It's not restricted to in any way. You can use it however you want. You can use it commercially. You can use it. You can change it. You can put it on your website. It is literally completely open. And here are some examples of uses that it's been put to, including the NIST and the FDA chose it as their sole source for standards for genomic and cellular data, and it's now international in many countries. So I urge you to take advantage of it if you need data or cells for testing therapies. For example, that's my brain there, brain slices from 2009. A new set was done just this summer in Toronto. Clearly, they're not actual brain slices, or else I would be less coherent. But anyway, it's your project, and you can volunteer for it, or you can just make withdrawals. So I'm looking forward to the discussion that we'll have later after the other speakers as part of the panel. Thank you very much.
Well, thank you, Dr. Church, for such a provocative and sort of visionary talk. And for those of you who are in this field, there's so much rich genius and complexity and science behind all those slides. But I also know at the same time that there's a lot of question in this audience. You know, what's the role of technology to slice and dice and, as some people have said, to play God in terms of shaping people's health and destiny? And if it's okay, I'm just going to assume it's okay to share our discussion on the sideline to put this in the context. I was talking with Dr. Church and Dr. Chisholm, who's here from MIT, and we were talking about supplements. You know, what supplements do you take? And the notion was, yeah, these may help. But what Dr. Church shared candidly was, if we really want to go for the end goal and to sort of reduce suffering, and those are my words, and extend life and health and quality, he thinks that we need to really get in there and change the code. That's going to be the most direct path. And so what I want to share is this sort of yin-yang complexity between compassion and empathy for health and life and technology, the soft touch and the high tech. And I think that's sort of the spirit of this talk today. So with that, we're going to move on now. And it's my pleasure to introduce our panel organizer, Dr. John Denninger, who's going to coordinate the next three presenters and then facilitate a discussion, including Dr. Church and the three presenters at this table. Dr. Denninger is a friend and colleague. He's a director of research. He's my counterpart at the Benson Henry Institute for Mind-Body Research at Mass General Hospital, and he's also an instructor in psychiatry at the Harvard Medical School. And his own research is at the forefront of understanding the genetic and epigenetic impacts of things as opposite as these manipulations, mind-body therapies. So please join me in welcoming Dr. Denninger.
So thanks, Peter. Thanks, Dr. Church. That was incredible. I'm going to take out my notes so I know what to say. That's always useful. How's everyone doing? Good. Fantastic. All right. So Peter just said it, but I'll say it again. So I'm going to talk for just a few minutes. We're going to bring up three speakers. Then afterwards, the three speakers and Dr. Church are going to sit there, and I'm going to moderate the questions that you guys are already entering on Slido, right? Lots of really exciting, interesting questions, yes? Good. Fantastic. Keep going with that. All right. So what we're going to be doing in these three talks is essentially taking like a little walk down biology. We're going to be starting with genes and DNA, DNA, of course, is where genes live. They live in the nucleus, just to remind people. And genes come in different alleles. People are familiar with that concept, right? Different alleles. It's like back in high school biology, you may have learned some, like a gene for blue eyes or something like that. And you might inherit different alleles from your two parents. Well, that's why Dr. Church was mentioning the importance of having genomes that keep intact the information of which gene came from which of your parents. Our first talk by Catherine Hall is going to show the kind of gene-based precision medicine that's being used in things like cancer treatment, and showing that can also be used to understand how vitamins and supplements and even food are interacting with our bodies. Our genes influence which foods and vitamins are good for us, and maybe even which ones are bad for us. After that, we're going to turn to changes to DNA. So changes to DNA in the sense of not changes to the DNA sequence. You guys know that for the most part, you're born with a DNA sequence, right? And that exists throughout your lifespan. These are changes to the structure of DNA using chemical moieties like DNA methylation. So this is what's called epigenetics. Have people heard that term, epigenetics, the epigenome? Olivia Okereke is going to be talking about that, showing that even with the genes you are born with, lifestyle and stress can lead to epigenetic changes which regulate ultimately how genes get expressed. Does anyone remember what messenger RNA is? People in the room know what that is? So messenger RNA is essentially what gets expressed from genes. If a gene gets turned on, the first step in the process that leads to the production of protein is messenger RNA. That starts a cascade that then leads to, I'm a protein biochemist, so it's sort of my favorite thing, leads to the production of proteins. Proteins they're not just things you get in a powder and eat if you're trying to build muscle. Proteins are things like specific molecules that do specific functions in the body. Things like hemoglobin. Have people heard of hemoglobin? Hemoglobin is the protein that, of course, exists in red blood cells. It's the thing that makes them red, and it's the protein that carries oxygen for our bodies. Manoj Bhasin is going to be talking about how mind and body practices like meditation and yoga and the like, turn genes on and off that are involved in the immune system, in inflammation, and downstream from that are important for risk of illness. So a few things I want to keep people in mind. These are going to be short talks, by the way, very quick. So you have to pay close attention while people are talking. But some things I want you to keep in mind while these talks are going on. One is that the interaction between genes and environment is always a two-way street, right? There is interaction on genes and how they are expressed from the environment, and there obviously also is influence on how we respond to the environment based on our genes. Precision medicine, which can be amazing for things like curing someone's cancer in an individual's specific cancer. That kind of approach also applies to integrative medicine approaches as well. It's not unreasonable to think that a certain supplement or a certain kind of practice might be very specifically important for a given individual, depending on what their genetic background is. And then finally, I just wanted to say that sort of all these biological levels that I've been talking about, from DNA to the epigenetics to messenger RNA to the transcriptome, they are all addressable by what we choose to do as human beings. What we choose to eat, how we choose to live our lives, whether we smoke, whether we exercise, whether we engage in mind-body practices and the like. In a lot of ways, it's sort of like the brain. What's the brain for? I'm a psychiatrist, so I like the brain. The brain is there to integrate what's going on in the environment, right? The brain allows us to take that information in and use it to change our internal milieu. making sure that that internal milieu can be appropriately adjusted. Lifestyle choices that are encouraged by integrative medicine interventions can profoundly affect that milieu. That's the bottom line of what I think we're trying to incorporate here. And I just have to say that it's pretty incredible that the Osher Center has brought together this group of people to talk about these incredible ideas. All right, so the first person to talk is going to be Catherine Hall. Catherine is an assistant professor of medicine at Harvard Medical School. She's an associate molecular biologist at Brigham and Women's Hospital and director of the placebo genetics in the program for placebo studies based at BIDMC and Harvard Medical School. So Catherine works on the pharmacogenomics of placebos and supplements with an emphasis on aspirin and vitamins in cancer and also in cardiovascular disease prevention. So she's going to be starting us off on that path I was talking about before, starting with DNA and genes. She's going to be talking about how different alleles of genes can determine how much vitamins and other supplements can be helpful or even potentially harmful for your health. And what is that? That's precision medicine. Please welcome Catherine Hall.
So Dr. Church gave us a really wonderful futuristic view of how we're going to be able to change ourselves, make ourselves better, stronger, faster from within our genome. But everybody here sitting in this audience, including myself, we're stuck with what we have right now. And we have actually, if you think about it, half of US adults think about this problem. How are they going to stay healthy? How are they going to prevent disease every day? And every one of us turns down that aisle at CVS or Whole Foods with all those vitamins and supplements, and as soon as you turn that aisle, you become your own pharmacist. You become your own physician. And you have to make decisions about, are you going to swallow your vitamins? Are you going to chew them? Are you going to, even now you can vape them? And you have to make a decision about, which combination you're going to need. Think about it. You're going to put vitamin E, vitamin C, a multivitamin, and let me get a supplement. Let me get some fisetin, some quercetin. And we make these crazy cocktails every morning when you take your vitamins or at night. And really, we're going on what the label says, what our friends say, what our parents did to us when we were little, gave us our chewy vitamins. And for the most part, we're going on what the media says. And if you listen to the media, things really go back and forth, because the bottom line is that even though half of Americans use vitamins and supplements, we don't know if they work. And we don't know if they work, not because the animal studies haven't demonstrated really amazing things where the antioxidants protect, as Dr. Church intimated, antioxidants protect from aging, can prevent disease in animals. And we've worked out these elaborate biochemical pathways that are very compelling. And we've done observational studies where we've asked, people who have used multivitamins or vitamins or antioxidants whether or not they're healthy, and we've shown that actually when you do these observational studies, that actually there's a benefit to some people for using these supplements. But when we do clinical trials, we don't get a difference. Here's a really large clinical trial, 40,000 women, the Women's Health Study. They were randomized to vitamin E or aspirin for 10 years, and there was no difference at the end of those 10 years in rates of cardiovascular disease or cancer. So why do vitamins fail to show efficacy in clinical trials? Well, that's a question that we are very interested in, because I think we all kind of know deep down, and that's why we're still taking them, that they do work for some people. But who do they work for? And so that's essentially the question that I've been asking, and we've zoomed in on several genes that we study, and I just want to walk you through what we know about one of the genes. And this gene I'm going to talk to you really briefly to give you a sense of the complexity of this question and the potential of this question is COMT, or catechol O methyltransferase. And that's a fancy name for a protein, and we've heard about proteins, they're coded by genes. That protein basically takes this CH3 group in the top right here. Let's see if I can get a cursor. That's called a methyl group, and it sticks it on catecholamines. And this is really amazing because catecholamines are dopamine in your brain. Epinephrine and norepinephrine make your heart beat, important for fight or flight. And catechol estrogen, which is a breakdown of estrogen. And estrogen is really important for many things, our reproductive health, but also in protecting us or increasing our risks of cancer. So here's one enzyme that can modify catecholamines that work across multiple systems. So what happens when we have genetic differences in that one enzyme? Well, I work on a polymorphism that is called RS4680. That means that at one locus, one place in the gene, a G gets changed to an A. And that might not seem like a big change, and in fact, there are 500 million changes that can happen or more in the body because you're constantly trying to put together all those little bases that make up all those A's and C's and T's and G's that make up your whole genome. So we're bound to make mistakes. Sometimes it's not a mistake, sometimes it's intentional. And when you get a G that changes to an A, the activity of this enzyme is decreased threefold. And what does that look like? So think about it. If I have a very active enzyme that's breaking down my dopamine very efficiently, after I eat that chocolate bar, it's over for me. I'm going to now, my dopamine is broken down, so I need to maybe eat another chocolate bar or maybe move on to something else. So if I have high activity COMT, I have less dopamine. I also have less epinephrine, less norepinephrine. I'm probably less prone to stress, as opposed to if I have the low activity allele. I tend to have more dopamine, more epinephrine. I experience more pain. And there's been an amazing, this is the most popularly studied polymorphism in psychology. And because of that, we know a lot about the types of people that have this polymorphism. Remember, this is one gene, but it has some very interesting effects. And I just want to show you what we found in the women's health study, because we asked the question, because we knew that COMT was associated with placebo, what happens in the placebo arm of the women's health study? And I'm going to show you what happens. In these black lines are what happens to the people in this study, in the women's health study. The met women on placebo had the most cardiovascular disease. The way this graph works is it's the 10 years of the trial on the x-axis and the rate of cardiovascular diseases on the y-axis. And if you're high on this axis, you have more disease. So the met women had high rates of cardiovascular disease on placebo, but when they took vitamin E, their rates were significantly better. These women benefited from the antioxidant supplement vitamin E, as opposed to the other women, the high-activity COMT people. They did better on placebo, but when they took vitamin E, their rates of cardiovascular disease increased. Now, we've looked at this in several other studies. We've looked at what happened with aspirin in that clinical trial. We saw the same thing. We've looked at clonidine in a trial of chronic fatigue syndrome. We essentially saw the same thing. On this screen are tolcapone, which is a drug that targets COMT for Parkinson's. So basically, they're trying to basically block COMT from breaking down dopamine, so there's more dopamine available to Parkinson's. But think about it. When you start messing with COMT, you're messing with dopamine, epinephrine, norepinephrine, catechol estrogen. This is complex stuff. We also know that COMT breaks down paroxetine, Paxil, and it breaks down phenytoin, which is a drug that's used for epilepsy. So the bottom line here is that this is just one gene with many drugs. And here's the kicker. When you walk down that aisle at Whole Foods this evening, tomorrow, this weekend, remember, you're going to be crossing many, many drugs that are metabolized by COMT. You need to know your genotype. Obviously, we need to do more to test this and to validate it, but when you see quercetin, fisetin, S-adenosylmethionine is the donor, is the molecule that's giving the methyl group to COMT, that's sold in Whole Foods. All of these drugs are catechols that are degraded by COMT. So my take-home message is diseases are complex, genes are even more complex, and the drugs that we use to treat them are doing multiple things, and we really need to start digging into what genes, what drugs, what diseases, and how all these things interact. Thank you.
Thank you very much, Catherine. That was outstanding. So next we have Dr. Olivia Okereke. So Olivia is an associate professor of psychiatry and an associate professor of epidemiology at Harvard Medical School and the Harvard Chan School of Public Health. And she's director of geriatric psychiatry at Massachusetts General Hospital. A fun fact, Olivia was one of my chief residents when I was a resident in psychiatry at MGH McLean. Olivia works on ways to identify what we would call like modifiable factors, things like diet and exercise, which are involved in adverse mental aging, which I think I'm going through daily now. She's also working to take that understanding, to take that research and integrate it into strategies for large-scale improvements, sort of public health level improvements in late life depression and cognitive decline. And I'm just hoping that she finishes all that work before I get a lot older. That's my wish. So Olivia's talk is going to take us further down the trail I was talking about before, moving from genes and alleles to epigenetics to changes in things like DNA methylation, showing how changes in lifestyle can alter the structure of DNA in a way that can control how genes are turned on and off. Please welcome Olivia Okereke.
Well, first of all, I just want to say thank you to Dr. Wayne and everyone at the Osher Center for this opportunity, because the work that I'll be describing actually was funded by the Osher Center, was supported by the Osher Center. So I'll be talking about how stress relates to biological aging, and the way that this came about for me, I was very interested in the idea of how biological aging could be a readout of the impact of stress on the body, as stress is one of many modifiable factors of interest. So, for example, we can think about the telomere, which is something that I've been looking at over the years, and there have been a number of studies that have shown relations between depression, anxiety, psychosocial stress, and modifiable lifestyle factors like cigarette smoking or exercise on telomere shortening. And the mechanisms hypothesized involve, for example, increases in inflammation and oxidative stress, which may precipitate accelerated telomere shortening or telomere erosion. And given the fact that part of the role of the telomere is actually to protect integrity of DNA, genomic stability, there's also this interesting potential for feedback where telomere erosion accelerated can lead to increased DNA damage, which may set up consequences for worse mitochondrial function, which spin off more reactive oxygen species and greater oxidative stress, and there can kind of be almost a vicious circle, if you will. So this has been something that's been a tremendous area of interest with regard to the telomere, but I had a lot of interest in sort of looking at some other biomarkers of aging, and that's kind of what led to this study looking at epigenetic aging. So the particular vehicle for this is DNA methylation age, or what's been known as an epigenetic clock. So briefly, this was developed by, the particular clock we're using is not the only one available, but this was developed by Dr. Steve Horvath using an elastic net regression method to identify 353 CpG dinucleotides, which are these epigenetic DNA methylation marks on sites across the genome that uniquely predicted with a high degree of correlation, high degree of accuracy, chronological age. So the first question was to look at, well, how does epigenetic age in our sample relate to chronological age? Does it relate to lifestyle and stress markers? And how well does it relate to some other markers that presumably also may reflect biological aging, like the telomere or mitochondrial DNA copy number was another one we were looking at. So we're looking at how they relate to age and also how they relate to each other. So to do this, we leveraged actually a study, as Dr. Dannenberger was referring to, and also Dr. Hall, which was a vitamin trial. And this trial has been in the news recently. It's called the Vitamin D and Omega-3 trial. And I've been running an ancillary study that's going to be, as we finish crunching the numbers, looking at the depression endpoint within this vitamin D and omega-3 randomized trial. But as a subset of that, we have a group of people who are very well characterized in terms of a wide array of lifestyle, behavioral stress, psychological measures, cognitive function, and they're midlife to older adults. adults. We had cases who had a history of psychiatric illness and controls who did not. And we looked at these biomarkers within them using blood samples from which we extracted DNA. DNA methylation was measured using the Illumina methylation EPIC platform, and telomere and mitochondrial DNA were measured using PCR assay. And so what this slide illustrates is essentially that we see a correlation between DNA methylation age on chronological age that is quite strong, close to 0.9, and this is consistent with what other people have found. It's a very tight correlation. We see, as we would expect, an inverse correlation of shorter telomere length with greater chronological age, but we don't see a correlation for mitochondrial DNA copy number. So we're also interested in looking within this set how these biomarkers relate to each other. And surprisingly, there haven't really been many studies that have looked at all three of these intercorrelated. So this pilot was a really nice opportunity to try to get some sense of that. And we see a little bit of a relationship, an inverse correlation, as one might expect, between higher DNA methylation age and shorter telomere length, but not quite reaching statistical significance. We didn't really see the relationship for mitochondrial DNA copy numbers, so it doesn't seem to relate as well as the other two. But moving forward, we really wanted to get a sense of how epigenetic aging is related to the modifiable lifestyle factors. So setting up a situation in which we might think about, well, what are some changes we could make, for example, in health and behaviors that might portend lower epigenetic aging. So in looking at DNA methylation age, telomere, and mitochondrial DNA copy number, the bottom line is within this relatively small sample, of course, it is a small sample, we really didn't see an association for the telomere or the mitochondrial DNA copy number. And admittedly, this may have to do with just measurement precision and needing more people. But it turned out with DNA methylation, there was enough precision that we were able to see correlations with alcohol, with there being essentially higher DNA methylation age with alcohol, and a trend for smoking and BMI, and significantly inverse association of lower DNA methylation age with exercise frequency and metabolic equivalent task hours per week as total energy expenditure. But moreover, looking at psychological and stress factors, we found interesting associations here as well. We use a measure called the PHQ-9 of depression, where we didn't really see anything significant in terms of DNA methylation age or accelerated aging, but we saw a trend of a positive association. We used a metric of accelerated aging that's been developed by Dr. Horvath, and we found that actually higher anxiety was significantly associated with higher accelerated aging. So essentially, it's kind of an equation that maps out the extent to which epigenetic aging is greater than expected compared to chronological age. So it's giving us a sense of whether there's faster or accelerated epigenetic aging. By contrast, psychosocial support, which we measured as frequency of contact and quality of contact of psychosocial contacts, was inversely associated with DNA methylation age and measures of accelerated epigenetic aging. And also intriguingly, we found that people who had higher cognitive functioning had lower epigenetic age, and age acceleration, using a very high-quality measure called the Modified Mini-Mental State, or 3MS. So all of this has been very intriguing for us in terms of understanding the potential for these lifestyle modifiable factors and stress-related factors to relate to epigenetic aging. But a larger question, of course, that comes up is, this is a small study, it's cross-sectional. And, you know, right here in Boston, we're having the Gerontologic Society of America annual meeting, which is where a lot of these topics are being discussed. And it's actually noted, and there's been a bit of a consensus among folks, that we really need longitudinal data for this. We really need to get a better sense of how these measures of epigenetic age track individuals over time and what it's really telling us about how targetable they are. So using something like the vital depth might be a way to do that, and to try to understand not only influences of these vitamins and supplements on the target of epigenetic aging, but how they interact with baseline stress and psychological symptoms. And again, with great gratitude to the Osher Center, this pilot data was enabled us to get funding from NIH that just came through last month to actually address that in a much larger sample within this vital depth cohort. So I think I'll stop there so we don't run out of time, but thank you very much.
Thank you very much, Olivia. And so Now we're going to have our last talk before we do the panel. Continue to enter questions on Slido, especially while I'm talking and nothing important is happening. So Dr. Manoj Bhasin is an assistant professor of medicine at Harvard Medical School and director of bioinformatics and systems biology at BIDMC. He's also co-director of the BIDMC Genomics, Proteomics, Bioinformatics, and Systems Biology Center, and a senior investigator at the Harvard Medical School Vascular Biology Center. He's a close collaborator of ours at the Benson Henry Institute and has used really cutting-edge genomics and other omics technologies, along with systems biology approaches, to identify key pathways and genes that are associated with the beneficial effects of mind and body approaches. He's going to be talking about some of this work, specifically making the connection between mind-body practices and changes in gene expression. Please welcome Manoj Bhasin.
Thank you, John. Thank you, Peter and Helen, for inviting me here. It's an exciting set of speakers. So my lab uses cutting-edge genome technologies like what Dr. Church talked, basically, like where I think most of the technologies came out of his lab, like how we can measure all the genes at one go, like all 50,000 transcripts, which correspond to around 30,000 genes. How can we measure them to really see, like, how does these mind-body techniques are altering them? So this is the crux my lab do. So we use all of the technologies measuring the expression of all the genes, all the proteins, before and after mind-body intervention to really understand, like, how does these technologies really alter our genes. The first study we did is, and all this work is done in close collaboration with Benson Henry Institute, and we have like a lot of studies open up with them. So the first study we did is in 2008 and 2013, the repeat of that study, where we took a group of novices and trained them with mind-body intervention for 8 weeks, measured their genome 4 times at baseline and after 8 weeks, to really see what are the changes happening in 8 weeks. And then as a positive control, we added another group of individuals, which are the long-term meditators, which are doing yoga meditation from like 2 years to 10 years as a positive control. And we measured the genes at multiple times to really figure out, like, what are the changes which are consistent, not the random noise, because the changes due to these technologies are pretty small changes. So what we learned is that there are 2 kinds of changes in the genes due to meditation. One changes we given the name of progressive changes, means after 8 weeks you see a small change, but that change is much bigger in the long-term meditators. Like these genes like start low, they go a little bit high up. After 8 weeks, they go much up in the long-term meditators. And second kind of changes we observed is which are just in the long-term meditators. 8 week of meditation has no effect on those set of genes. And to really understand, like, what are these genes which are like progressively impacted by mind-body interventions, we performed like pathways analysis to really figure out, like, what are the biological things they do in our body, like how do they regulate different kind of processes in our body. So when we looked at the genes which are going progressively up, like these are the genes, like these are the novices, these are the short-term meditators, and these are the long-term meditators. You can see, like, they start from green and started going toward red. So like genes are going up. So if we look at the genes which are progressively going up, they involved in mitochondrial and energy production, electron transport chain, and insulin secretion, basically. So these are not random genes which are changed by mind-body intervention. They are really upregulating the energy production, electron transport chain, and insulin secretion. That is the first message we got from this data. Then the second is, like, what is the genes which are going down progressively by the meditation. So when we looked at those genes and do the same pathways analysis, what we learned is that they mostly belong to inflammation pathways, IL-7 and TCR signaling pathways. So they are all immune system focused pathways, mostly based on inflammation. So, but what is the key pathway? We still keep on going on our data analysis because these are multiple pathways, like is inflammation is driving it, IL-7 is driving it, who is doing it? So we did system biology because in our system, like all the genes work in coordination with each other basically. They are different group of genes. They talk to each other if they want to carry out a particular kind of process. So when we did the system biology analysis, what we learned is that the genes which are progressively upregulated, they are mainly focused into mitochondria. They upregulate the ATPase complexes in our mitochondria, the expression of ATPase complexes, and the insulin gene itself is upregulated by 8 weeks of meditation. And then if we look at what is going down, we see like MAP kinases, JUN, these are all the genes which interact with a transcription factor known as NF-kappa B. And NF-kappa B is the main gene that is responsible for driving inflammation, or very strong association. When you have a high level of NF-kappa B, you have a more amount of inflammation. So that is downregulated by meditation in 8 weeks. So then we carried out multiple other studies to really see is these hypotheses hold on. So we did a study in IBD, multiple myeloma. We did a study in hypertensive patient, PTSD subjects. So we recently published our hypertension study where we took a stage 1 hypertensive people and trained them with RR for 8 weeks and measured their blood pressure changes as well as measured their genome level changes. So what we observed is that 54% of the patients responded to mind-body intervention. Their blood pressure goes down significantly. Both the systolic and diastolic blood pressure, they become normotensive. And we call these group of people whose blood pressure goes down significantly as the responders, and the group of people whose blood pressure is not changing, we call them non-responders, and did the genomic comparison to really figure out what is different in these two groups. One group is behaving normal due to mind-body intervention, not the other group. So when we did the pathway analysis on this group, what we learned is that one of the pathway that is significantly going down in the responders as compared to non-responder is inflammation pathway. So this is if the inflammatory genes go down, maybe the blood pressure will go down. That is the hint we got from this data. And then we did the system biology on these pathways which are going down in the responders but not in the non-responders. And we again learned that one of the molecules that surface out as a key molecule for making all these genomic changes is NF-kappa B. So again, it's a driver of inflammation. And then we did a meta-analysis in which we looked through 21 different studies and try to understand these are some of the studies done by our group as well as the studies done by other group across the world, to really understand what they are observing. Do they observe same kind of inflammatory processes are down modulated by mind-body interventions or not? So what we learned is that one of the consensus that is emerging in the field is that most of the mind-body interventions, they down modulate NF-kappa B, the main transcription factor that is associated with inflammation. And this transcription factor is involved in lot of diseases, any chronic disease. You have a stress, any chronic disease, NF-kappa B levels goes up. And we have now evidences in mouse models, if the cancer has more amount of inflammation, it will have less resolution and poor prognosis. So if we can take care of this inflammation part by doing mind-body interventions, might be that will helpful in better management of the diseases. So that, I think, is what I would like to convey, that these mind-body interventions, they have genomic linkage, and they work through specific pathways in our genome, and they might be helpful in better management of the diseases by taking care of inflammation part of most of the chronic diseases. That's it. And I would like to thank my collaborators for this great amount of work. Yeah.
Fantastic. All right, so I'd like to invite all our speakers to come and sit at the panel. All right, let's get started. Is this it? That's not it. It's almost it. So I was just wondering, so Dr. Church, that was an outstanding talk, and obviously incredible work for us to hear about. You've heard from these guys here now. What are your thoughts about how the kind of work that many of us do in integrative medicine, looking both at things like supplements and natural products and practices like meditation and yoga and exercise. What are your thoughts on how those two areas kind of interact?
The simple answer is not enough. We need a lot more interactions to get integration. There's sometimes, I think, a fear on the technical side that there will be an easy answer that will make all their expensive work seem foolish, and fear on the other side that the technologists will ridicule things that have been around since our ancestors were pre-civilization, and everything in between, all possible. And I think the thing is we need to work together. I mean, it is true that within supplement, and we saw beautiful examples of Vail versus methionine in Compt, but almost everything you eat could have some positive and negative consequences. And when you have them in pure form, there's even more opportunity for mayhem. There are some nice big literature now on almost every category of supplement can cause cancer or enhance your risk for it. And some of that's going to be, there's going to be some combination genetics and environment and nutrition. But some of them are going to be hard to handle, even if you had perfect knowledge of genetics, environment, and nutrition. And there'll be some things that, even though they're hard to do that way, they're possible to do some other way. So for example, there might be some general thing that solves aging, which deals with a whole lot of complex diseases, which are hopelessly complex from an environmental and genetic standpoint, but could have, some relatively simple medicine approach.
Fantastic, thank you. So, am I going?
No, we're going to just have you use this computer for the questions.
Oh, okay.
And then you can just pick through these.
Okay, great.
Those are some of the top ones, and then we'll pick and choose.
Got it, okay. So let's see. This may take longer because of my brain processing speed. So I'm gonna, no, I can read them. That's not the issue. I wish that were the issue. That would have been good cover. I can't see them. That's the problem. So here's a question. The most popular one. How many genes would you have to edit, and how much would it cost to make up for the oxidative damage from a bacon double cheeseburger? I guess that's more.
That's a good integrative question that will require lots more research. I think the thing is there are going to be some things that are going to be personalized, and there's some things that are going to be generic, and there may be surprisingly a small number of genes that gain tremendous generic effects. So in mice, we can see a factor of 2 in longevity with 3 genes. That doesn't mean the same 3 genes will work in humans, or even that there are 3 genes that will work in humans to achieve a factor of 2 in longevity, meaning healthy, youthful longevity, not just being sick for hundreds of years. But it doesn't mean it's impossible either. I mean, we have proof of concept that you can do it with a small number of genes. So.
Yeah, no, very good. So this, I suppose, may be a question for a couple of folks on the panel, but there's a question about the effect of the microbiome in reversing aging. Anyone want to comment on that?
I'm really not trying to dominate the conversation. I do work on microbiome. I think it's still early stage. There are a large number of not just microbiome diagnostics, but microbiome therapeutics. I think it's safe to say there's no well done. All these things really merit randomized clinical trials, and there's none that I know of that does reversible aging in humans with microbiome, or for that matter, anything else.
Yeah.
There's some that are in progress, but none yet. None really yet, yeah. It's a long shot.
So I think not on mind-body side, but my lab works on other side, like where we look at the metabolites in your gut. So one of the metabolites that is produced by microbiome, we observed that IPA has some association with aging and like more incidence of chronic diseases, but I think that is a very small study. I think nothing is like that it is IPA is, low amount of IPA is causing the disease. We don't know that. But I think microbiome definitely play a role in altering our genes. So like bigger studies will answer, I think, concretely, like it can reverse aging or not. Yeah.
All right, this is a question for Catherine. So the questioner says, as a supplement taker myself, do you suggest that I toss my supplements today?
I didn't get to tell you the numbers, but about 25% of people, let's say if you just look at the COMT gene, because it's not the only gene, it's just the one that we've looked at in the most depth. But if you think about that one, 25% of people seem to benefit and have a protective benefit, and another 25% of people seem to have the opposite effect, and then there's 50% of people that are in between. And that's the kind of distribution of the gene in the population. So I think that we innately know what works for us, and I think that's why vitamins have worked for so many people, is because you go down the aisle and you kind of know that this, I have a friend that will never take aspirin. Well, she has a Val genotype, and in our studies, aspirin is not, a good thing for her to take. She just knows it. So I think that till we have more evidence, I think you have to trust your gut.
Yeah. So this is a question for everyone, and I think is a very important question, which is, based on the research that you all do, what needs to change about the way in which we educate the next generation of healthcare providers? What do we need to be teaching people now so that they will be ready for the kinds of changes that are afoot?
Sure. So, I can reflect on what things were like, certainly when I was in medical school, and we didn't really have much education on nutrition, which was pretty shocking, given how important that is for the outcomes that have such a global impact on disability and quality of life. It got to the point where a group of folks got together and said, we're going to demand a nutrition module be added in our second year so that we'll get some of this. I think things are a lot better now, but there's no question that the area of prevention, as Dr. Church pointed out prominently, needs to be really focused on among healthcare providers, and that is something that there's some improvements in that regard. So, for example, CMS reimbursing certain kinds of preventive screenings being done as a way to incentivize healthcare providers doing more of this. But more broadly, greater education among healthcare providers about complementary and integrative methods as a way to achieve health outcomes. That it's not just about prescribing pharmaceuticals, et cetera, and just sort of creating more of a culture around people appreciating the value of that.
And so, does anyone else have any comments on that?
No, I think, we definitely, I think, pharmaceutical are great, basically, but I think they are not answer for everything. So I think adding these adjunct modalities like yoga, meditation, mindfulness, might help the patients to better manage their disease, to give them a better handle on their disease. Because I think there is as we heard from Dr. Church talk, basically there is it's a great time to have a personalized medicine, like where you manage your disease according to maybe like your genetics and other social factors that are playing role into it. So I think I will say like the combination of personalized medicine on the pharma side, along with the integrative medicine, will be a great combination. Yeah, but that's future, not here right now.
I would just add, even after it took a while for us to get nutrition on the medical school education, it's taking even longer to get personalized nutrition on the medical school education, or personalized anything for that matter. And just because something is currently used generically and is currently used without randomized clinical trials doesn't mean that it's exempt. It doesn't mean that there aren't individuals who respond poorly to certain kinds of athletics or yoga or meditation or nutrition. We really need to have personalized randomized clinical trials. I know they're expensive, but if you think education is expensive, try ignorance.
Right. Well put. So there are actually a couple of questions here asking about the question about sort of equitable distribution of these kinds of technologies worldwide. How do we make sure that it's not just people in countries like ours that benefit from these things? How can we make sure that stuff gets equitably distributed? That's it's a small question. Please solve the world's problems.
I think now the technology, like if I talk about genomic technology, I think they are becoming pretty cheaper modalities. And I think like now we have a $0 genome basically. And I think any lab, like my lab, like we regularly do like $600, $700 genome. And I think all the countries, I think across the world, like if they cannot do like a whole genome, maybe like if like we are talking about inflammation is impacted by these mind-body interventions. Can we measure like these 5-10 genes? That is like very minimal amount of money. And I think technologies are available in every country, I will say, like for doing it. Yeah, I think these are not like technologies which are just developing out and here, and we can only use it here. I think every country can use it, I think. Yeah, they're pretty standardized.
There are very few technologies that are equitably distributed. One of my favorites is smallpox immunity. That was a major worldwide plague. Now, literally everybody has access to it. You do not have to take a vaccine. Even the vaccine is inequitably distributed. But once you make it extinct, then you don't even need to distribute vaccines anymore. So that's a rare example. I think having a $0 technology, as much as I made a big deal of that, that's not adequate. We had $0 seatbelts for a while where they were installed in your cars and people sat on them. It was against the law, but they sat on them. So $0 in laws is not enough. You need to have an annoying sound that's made.
So here's another question. So what is the gene-based evidence for the possibility of passing on the consequences of our positive behavioral health changes, mindfulness, compassion, et cetera, from one generation to another? That's a great question. That's really an epigenetic question. Right. Well.
Yeah, that's definitely a very compelling topic within epigenetics, and I'll certainly let others chime in as well. But a big part of why we like to focus on aspects of stress or mind-body is that these are things that are under individual control. These are things that people can modify. And the possibility is that, for example, if you look at one generation, not only could you see maybe epigenetic changes that are transmitted to a second generation, but it's possible that because these changes are heritable, that you could even observe it in the subsequent generation, or what we call like F2, in that generation, so that if there's some either detrimental, like starvation, famine, war, exposure, or a more positive one, that consequence may be transmitted epigenetically. So that is something that a number of people are actively researching. We certainly need a lot more data on how this works, but there have been some well-publicized examples of how this might work.
Excellent. So here's another big question that I suspect we're not going to solve right this morning, but what are the chief bioethical issues in genetic engineering today? It's a provocative question.
Well, we have addressed some of, like equitable distribution is one of the ones that floats to the top of the list. Germline is another one that tends to be significant. The National Academy of Sciences described circumstances under which germline editing could be of significant benefit, medical benefit, where the benefits outweigh the risks.
Dr. Church, can you just briefly explain to the audience what that means?
Germline just means that you've edited either the egg, the sperm, or the embryo such that whatever edits you've made go on from generation to generation. The main reason that's initially raises alarm is the idea that you're going to make something new, but most of the gene therapies that we're dealing with are could be to just reverse it to the standard allele that is most common throughout the population, very well tested. So it's not like you're making something that's untested. You're making something that's extremely well tested. It's just you want to make sure there's nothing off target. You know, there's a lot of these so-called ethical issues. Once you address safety and efficacy, which is sort of the standards of FDA, they diminish in their concern. There are other things that the FDA does not deal with, and one of them is cost, and the other is extremely long-term unintended consequences. That's not their purview. That's not their mandate. So I think most of the issues come up around those, and as well as reflections and significance for religious and other societal and personal views can be swept under the rug. You can commoditize your relationship with your children and so forth. Yeah, so there are some issues unresolved.
I think just to add to that, in as much as the West is innovating on all of this, I think we are the guinea pigs, and it might even be too soon to worry about equity, because we want to make sure that if you want to share technology, that it's going to benefit people. And I think we're still, as you said, understanding the short-term and long-term consequences of some of this technology. I think, there's a recent fascinating story with even the CAR-T technology where it had amazing effects on that young girl that we saw, and then as it got, broadened, it had some unexpected consequences. So I think we're still learning, and I think we have to be cautious who we roll out what to and when.
Great. Thank you.
Yeah, if cell phones had exploded our brains, as some of us worried, it would not have been a big gift to make that equitably distributed immediately.
Well, it's quite clear that we've exported a lot of things that are not great gifts to many other parts of the world. So this is I think a very key question, which is, for healthcare providers, when is it? How long is it going to be when we're going to be starting to use some of these things clinically? Looking at DNA methylation, looking at epigenetics, looking at transcriptomics. Any sense?
Like, if you, for integrative medicine, I think we are a little bit far, but I think if you look at personalized medicine, cancer treatment, it's already, I think, ongoing. People are doing personalized tumor sequencing to really figure out what are the drugs going to work for them. So that's already in reality going on. And now I think with some of the technology companies, genomic companies like 23andMe, which are selling these kits in $50 or $40, tell you okay, what are the genetic variants you have. So I think the technology is expanding, but for integrative medicine part, I think we are a few years away. But I think for cancer biology, I will say cancer treatment, it is already there. Yeah.
And are they looking at epigenetics as well? I actually don't know that.
Yeah, for diagnostic purpose, some of them are looking at some markers which can diagnose cancer, but not for the treatment purpose, I think. Yeah.
There certainly are other examples within our own field of psychiatry. It's actually becoming increasingly popular to use these pharmacogenomics kits, which actually have proven quite helpful in many cases at low cost to more strategically identify treatments that will work in terms of antidepressants, for example, because it's really kind of a, throw the dart process in many cases for many patients, and it turns out that type of screening, looking at the pharmacogenomics profile, can help this be more efficient and lower the time that it takes to get someone well on the right medication. So I think something parallel to that, if it were to be scaled up using these kinds of technologies we're talking about, would maybe be a nice application.
So, Manoj, you mentioned 23andMe. There's actually a question here about the panel's views on this kind of commercial testing.
So I think 23andMe or ancestry data, like if I say basically, I think that can be the data that you can have, but I think how much clinical implication that data is going to have, I think is still a little bit away, because I think our healthcare providers might not be that comfortable in using that data. But I think you can have that data, and if you want to say okay, someone is suffering with the family has a cancer history, so you might use that data to figure out if you have some kind of mutation profile that is making you prone to cancer. But is healthcare providers are going to use that data? That is, I think, still a outstanding question, I think.
I don't think the problem is the healthcare providers adopting the ancestry data. I think the problem is the ancestry data is poorly predictive of medical outcome. I think what's predictive of medical outcome is the whole genome sequencing. It's not perfect, but it's at least aimed at medical outcomes, and it includes all the serious Mendelian diseases that are actionable either by genetic counseling or in some cases by preventative medicine today. Everybody is at risk for it. You don't need to have a family history to have action on that. And so there the barrier is not that it's half empty, half full glass. At least it's half full. Well, the ancestry is so far from medicine that it is actually quite distracting, in my opinion. I mean, you're analyzing 1 million sites out of 6 billion, and they're the million sites that are least likely to cause serious disease because they're so-called common polymorphisms. That means that both versions of them are very common in the population, which means they're not typically sick, either one of them. But there are millions of positions which are very sick. They're Mendelian diseases. 5% of our babies are born with that. So I think there's almost no real barrier to using those in medical clinics for dealing with Mendelian diseases, which are very, very serious and are not handled by those methods. But there are offerings. I mean, the $0 and the $1,000 genome are genomes, and those are available.
I'm going to ask you for a prediction. This is my question. How long is it going to be before, say, in the United States, everyone's genome is just done as a matter of course and it's just part of their medical care?
Well, I'm fairly sure you don't mean everyone, because everyone doesn't drive cars yet or have televisions.
Most people.
It's hard to say. I mean, if you had asked me in the late 80s when most people were going to have computers, it could range anywhere from tomorrow to never. And then suddenly in 1993, basically everybody adopted them in about 1 year, and now you can't pry them loose from our hands. I think there will be a year where there will be a gigantic tipping point. I used to think that magic would happen when we hit $1,000 genome, and I quickly, before we got to 1,000, I realized it wasn't low enough. Then I thought that maybe $0 would be low enough, and I'm not sure that that's going to have an impact. I think people have to see that it is, we have to communicate that it is valuable today. It's not something that's valuable 10 years from now, otherwise they'll wait 10 years. And it is safe, that that the insurance companies are not going to get access to it. And that's why at Nebula we put so much emphasis into encryption. We can encrypt it so well that even you don't know your genome. And so if the insurance company comes for it, they can't hack it. In fact, it's more likely they'll hack it by taking it from your hair or from the dust that you left on the table than they will hack it through an encrypted database that you own on your computer. Yeah. Anyways.
All right, one final question, and that is, it disappeared, the one I wanted to ask. Oh, no, here it is. Just briefly, for people out here who are clinicians, any brief headlines to share on the connection between mind-body therapies and genomics, broadly speaking? Manoj, I know you have one.
Yeah. So I think definitely, from our studies, we see that mind-body therapies, they impact the genome, and this is not a random impact. They really work according to all the studies that we have done and others done is inflammation pathways. And if you think inflammation is maybe one of the things you want to handle for helping your patients in better disease management, I think definitely there is an impact of mind bodies on your genomics. Yeah.
Anyone else have a 10-word headline?
Yeah, I think that it's clear from, as a placebo researcher, that interventions that are not necessarily pharmaceutical-based can have amazing effects. I mean, just if we were to hear a fire alarm right now, all our biologies would change, our epinephrine would go up, and we would probably be ready to run out the door. So it's not news that you can change your biology with non-pharmacological agents. And I think what we need to do is understand and harness it and not be afraid of the power that's in all these different modalities. And I personally love the genes that I study because I think there's something to be said about going from the brain and dopamine to the body and epinephrine and norepinephrine. It's a really nice mind-body axis. So I think it's not, I mean, people get afraid when we start talking about molecules in terms of are we going to take away the special essence of what we are? But the reality is we are all of that. We are the molecules and we are the air. So why not embrace it all?
Fantastic. That's a great point to end on. Let's give a big round of our applause to our incredible speakers this morning.
So thank you again to Drs. Church, Hall, Okereke, and Basin for your excellent presentations and for your participation in, I think, what we were hoping, a provocative transdisciplinary discussion. And thank you to Dr. Denninger for your skillful facilitating.